through the meiosis section of your assignment. These questions down here at the bottom, they're really just the basics, the ideas that you need to be able to take away from meiosis. The first one is given to you. How do the number of chromosomes in a sex cell compare with that in the parent cell? So for example, human cells, we have 46 chromosomes. In somatic cells, but we have 23 chromosomes in sex cells. 23 is half of 46. You half the number of chromosomes in normal cells, parent cells, when sex cells are made. If the number of chromosomes in the skin cells of an organism is 28, what is the number of chromosomes in the organism's egg cell? This is just asking you to apply the answer to number 1 here. Sex cells have half the number of chromosomes of body cells. So this organism's sex cells will contain 14 chromosomes, half of 28. How many cells are produced at the end of meiosis? You can check the image diagram we have up here. 1, 2, 3, 4. 4 cells, 2 divisions. So you have interphase. And then the first set of PMAT happens. 1 division gives you 2 cells. PMAT happens a second time. Both of those cells go on to divide again, giving you 4 cells. In which phase of meiosis does crossing over occur? And what results from this process? So you can see in prophase 1 where the ends of these homologous pairs are touching, and when they do that, genetic information is exchanged. They swap genes, basically shuffling the genes that mom and dad have for all of the genes on chromosome 1 or 2 or whatever chromosome it is. So crossing over occurs during prophase 1. And what results from this process? Genes from homologous pairs trade places, and this leads to genetic diversity. Describe the activity of chromosomes in metaphase 1 of meiosis. How does this activity differ from the activity of chromosomes in metaphase of mitosis? Okay, up here in metaphase 1 of meiosis, what you see is a tetrad, means a group of 4, of homologous pairs of chromosomes. Line up at the center of the cell. In mitosis, wow, sister chromatids line up at the center. So I'm going to do a little dotted line. You can see where I'm leaving space for the next question. In which phase of meiosis 2 does the cytoplasm divide? Well, it happens twice. Cytoplasm divides first in telophase 1 in cytokinesis, and then again in telophase 2 in cytokinesis. So you can put telophase plus cytokinesis 1 and 2, and that's just saves you from having to write it out twice. Explain why mitosis could not provide for the sexual reproduction of offspring that contain the same number of chromosomes as the parents. Okay, easy way to answer this. Mitosis produces genetically identical diploid cells. Sexual reproduction requires haploid cells to unite. And that's the easiest way to answer that question. You could elaborate more if you want to. You can, but that's the gist of it. The process of mitosis creates cells that have too many chromosomes for them to be passed on to offspring through the process of sexual reproduction. Now for the backside. These images are drawn in different styles. We don't know what these phases of mitosis are going to look like when you see them on a test, or when you see them on the STAR EOC exam. So you're just going to have to get used to seeing them represented multiple ways. So starting here, my key features I should be looking at is there's a division happening, and this division is resulting in two separate cells. So if I wanted to check the images that I provided you on the other side, the image that looks most closely aligned with this is going to be this telophase 1/cytokinesis. So that's what I'm going to put. Oops. So in the next image, I see two cells. That tells me that we're going to be in meiosis 2. The first division has already happened. And in these two cells, the pairs of sister chromatids, homologous chromosomes, are lined up in the middle of the cell. So when I look, my hint is that they're in the middle. We're already in meiosis 2, so metaphase 2 is the best answer for that one. In my next example, I see the chromosomes condensing, but I don't see anything lining up. So what looks most similar to this? Probably interphase. This is the very beginning of this process, so we'll go with interphase. And then we have a single cell, and the chromosomes are being pulled to opposite ends. So I'll look at my picture, one that I gave you. It looks like those chromosomes are being pulled apart. So the best choice that we can make is anaphase 1. In the next image, we start to see this pinching in the center of the cell. The chromosomes are still being pulled apart, but now that we have this pinching happening, this tells me that I'm looking at one of the phases of telophase. Only one cell is dividing at this point, so this is telophase 1. Now that we can start to see some patterns of single cell with those chromosomes lining up in the middle, hey, this is middle is metaphase, single cell, oh, must be metaphase 1. No division has occurred yet. Down here, I have 2 separate cells, and it looks like the DNA is uncondensed. This is going to be in between meiosis 1 and meiosis 2. And that in between stage is called interkinesis. Inter meaning between. Kinesis in this situation means division. So it's between divisions. And then we have 2 cells where the chromosomes are being pulled apart. We think anaphase. If there's 2 cells dividing, then the first division has already occurred. So this is anaphase 2. And then our last example, there is pinching here in the middle of these cells, 2 cells at the same time. This is going to be telophase / cytokinesis 2. And in the last section, based off of the description, I'm deciding what phase that cell is in. So homologous chromosomes pair up and form a tetrad. Formation of a tetrad happens in metaphase 1. Tetrad means a group of 4. So both pairs of homologous chromosomes are lined up with their sister chromatids in the center of the cell. So much vocabulary. Spindle fibers move homologous chromosomes to opposite sides. What? We have to use vocabulary again. So because it's telling me that homologous chromosomes are moving opposite sides, that means we are still in meiosis 1. Moving apart, moving to opposite sides, that would give us anaphase 1. Nuclear membrane forms, cytoplasm divides, 4 daughter cells are formed. This is going to be cytokinesis 2, the second time the cells completely divide. Chromosomes line up along the equator, not in homologous pairs. That means they're just lined up with their sister chromatids. This will give us metaphase 2. Crossing over occurs. This is one that you're just going to have to know this. If you know any single stage of meiosis, know that crossing over occurs in prophase 1. Because crossing over is the most important thing that happens as far as contribution to genetic variation. Chromatids separate. Chromatids, that is here, the ones that are exact copies of each other. Chromatids separate in anaphase 2. Homologs line up along the equator. Guess what? That's metaphase 2 again. And the cytoplasm divides, 2 daughter cells are formed. That is cytokinesis 1. First division results in 2 daughter cells, and the second division results in 4 daughter cells. So that was the assignment over meiosis. Hopefully you found that helpful.